Um, this is a poetry art area of work, but he asked me to talk a little bit about David Baltimore and his work. I'm not a molecular biologist, but I trained in molecular cell biology. I'll try to keep it as simple as I can because he asked me to talk about David Baltimore and how we could inspire young people to come into science. Thank you to all the guests who talked about poetry, music, light. It's beautiful. Thinking about science and art, we always look at intersections because there's always curiosity. Like she was talking about how her life has been regimented and she's been trying to work around it to find a voice that is not regimented. And she sang such a beautiful voice, with a beautiful voice, this beautiful song. And I started thinking about a few things. As a kid, I always wondered, how would sun, disappear in the west and come back in the east? Going back to the idea of light in some way. And one of the beautiful experiments I think people have been trying to figure out for a very long time is the light particle or is it wave? I don't think anybody's ever figured this out. I mean, you're showing me this, how light bends, took me back to that classic experiment where people try to have, study this using something called a double slit experiment. What they would do is they would throw light through these double slits. When they look at the outcome, it would show as if it's particles. But when somebody is capturing that, it would behave like it's a wave. It almost makes me feel like if somebody is watching you, you tend to have a different result. Not sure what that means, but when you showed this, how light bends, took me back to that classic experiment where we could never figure this out so far. Thinking about music too, I thought about, what would be the primordial sound? There are infinite sounds that are out there, but we can only make music out of very few sounds, and we've captured them only in 7 notes. In all the cultures, we only have 7 notes, and based on that, we make music out of it, right? Thinking about, so many noises that are out there. Can we make music out of it? Maybe not. But what is that primordial sound that actually makes us make music out of it, right? So anyway, the point of all this is that both science and art intersect, and we're all trying to do the same thing. Like you are trying to find your voice out of your regimented contract writing. We're all trying to figure out what the truth is, perhaps, doing science. So what best way to talk about science, but to talk about these giants, David Baltimore. David Baltimore titled it as Life in Science. If you have not read this book, I would highly recommend reading his book. He recently passed away, so we think about him a lot lately. This book is called Ahead of Curve, written by Shane Crotty, and in which he talks about how young people being at the right place and the right time makes a lot of difference. He spent a lot of time at Cold Spring Harbor, MIT, and all the big institutes, but the biggest thing that drove him at that time was really asking the right questions and curiosity, perhaps. So to read it out, he said, the people who are going to make the breakthroughs by analogy with the history are going to be young, well-trained scientists working in unpredictable areas. So now, who is David Baltimore? So he's an American biologist, very popular. Most people know him. He's an academic leader, got a Nobel Prize, like he was talking about in 1975. He and Temin shared this Nobel Prize, and independent of one another, they discovered how the viral genome, what he was talking about, the RNA, is incorporated into human genome. That brings us back to this, asking this question, why should viral DNA be in human DNA? How did it even get there? I think that's the question I think they rather asked a long, long time ago. Now, why should we care? right? So just to put it in the context, humans have, close to 30 trillion cells. That is 30 million cells. That's about how many number of cells we have. And each cell is uniquely coded. So each cell has something called the DNA, which there are 6 billion nuclear codes per cell, which is the DNA. That's about how many we have. If you put it in the binary numbers 10101080 GCs, so you have 6 billion. Like, you can't even imagine in your head if you start counting numbers, you'll probably lose count after, like 10 or 12. So those DNA is going to transmit the message into something called an RNA, which stands for ribonucleic acid. Those are approximately 10 billion codes per cell, depends on how fast the cell is growing. And those cells eventually lead to the final signal, which is the protein. And you have approximately 5 to 10 billion per cell. So if you think about the hierarchy where all this starts from, it starts from a DNA, handed it over to RNA, and then a protein forms, right? So the question is it always so, right? So the point what it was called was something called the central dogma. So it's called a dogma because everybody thought that was the truth, and that was handed over to most of the scientists in like 1960s. But David Baltimore and others really asked the question, is this really the truth, and can we go back and re-ask this question? Now, how about a bacteria? So bacterial cell, we all know, it also has the DNA. It has close to 5 million nuclear codes per cell, and then that goes into the RNA and the proteins as well. Now, if you go to the next slide. If you now go to the virus, so virus is incredibly interesting. So now there are 2 different kinds of viruses. So one of the viruses is the DNA virus. You need the DNA to replicate itself, so you need the DNA, and that makes the RNA and the protein. And then you have RNA viruses too. So this is a different kind of virus, which has the RNA in it. And the big question is, how does RNA replicate, right? So we have a virus such as HIV, which started out to be a big problem around New York and all the other port areas. And then we didn't know how to treat it as physicians. And the bigger question is, does RNA replicate by itself, was the big question. And does RNA beget RNA? I don't think anybody really understood that question, and that was handed over as a question to David Baltimore and all others when they were very young, when they were in 20s. So if you were to, again, going back to thinking about the human central dogma, you have the human DNA going into the RNA, going into the proteins. But if you think about the viral DNA, right? So the viral DNA gets into your body, which is a form of an RNA, and that incorporates into the human DNA. So when it incorporates into human DNA, and a different kind of a protein is made, or a different, a protein is made, which is completely controlled by a signal of an RNA virus, right? So which is an incredible thing. What the thing, the questions they asked were, how does this viral RNA get into the human DNA? So during that questions, to most of the young people and people in the audience is this really serendipity or is this perseverance? So they took this question of how does viral DNA, RNA, get into the human DNA? Or is this, continuously doing it? Is this just luck or perseverance? I always felt like you increase the surface area of your luck if you stay, in a lab or keep doing what you're doing, right? So I don't know how many of you are familiar with this concept of black swan, which is, one-time event that ever happens in, a random event, which is 1 in a million times, versus where you're constantly going to the lab and keep on asking the same question over and over again, maybe 20 years down the lane, one fine day you will find an answer. Would you consider it as serendipity, luck, or would you call that increasing the surface area of luck? It's just because you kept on doing it over and over, and then that's what likes of David Baltimore and most of the scientists around here in the Harvard area do. So they discovered an enzyme called reverse transcriptase that converts the RNA from the HIV into the DNA. That's how it incorporates itself. So it almost questioned the central dogma and really discovering this, the paradigm shifting discovery that led them to a Nobel Prize. Not only that, right after they discovered that, they could find treatments for HIV, which is remarkable when people live as long as everybody else now who have HIV, and AIDS has more or less, been wiped out now. And the bigger thing is the advent of gene therapies, which is the newest thing right now, where you use these RNAs as delivery vectors into the humans so that they can treat people. So that's actually one of the remarkable things. And most importantly, I think vaccines and also cancers as well. So as I said, they got the Nobel Prize for discovering that, and they had a bunch of other important things in the science which kind of makes them unique. Thinking about life. So going back to introducing what they really did, like all of us, why we all get introduced to science or anything that we do, what we do is mostly young life experiences, right? And apparently David Baltimore went to high school like most of us, got interested in science, but his life apparently changed when he went to Jackson Laboratory, where he really learned the newer replication techniques, most of the techniques. He got really good at doing what he was doing, mostly technical. With that, he started asking really good questions there, because he has been interacting with, other Nobel laureates and all others. And that changed his outlook. And if I went back and read the book, and he almost made it sound like his life changed around the time when he spent time in Jackson Laboratories. And following that, he went to MIT, Cold Spring Harbor, and moved to Caltech. So in some of these pictures you can see where he was at Cold Spring Harbor, where he was trying to learn to do something called electron microscopy, where you would go in and really take pictures of subcellular, which is the stuff which is inside a cell. And him in MIT in 1971, these are the pictures I got from ahead of curve. And he was the one who founded Whitehead Institute at MIT here. And sadly, he passed away this month. So this is a tribute to David Baltimore. So this was his picture from his Nobel, after he was awarded Nobel Prize, again from the book. All right, so ending with something which I always felt was very important for most of us. Don't be afraid to be foolish. I think this is another Nobel laureate who said that, Phil Sharp. If you have an idea, just go with it. When I walked into this room, there were like 2 people, and I was asking, why is this guy doing what he's doing, right? And I think we're all foolish. I think we should all be foolish. With that, thanks for your time.